Welcome to episode 72 of the Reptile Gumbo podcast. It's been a very busy 2 weeks.
Yeah, it has.
In fact, I say I'm ready for it to slow down, but it's not going to. We just got back from, we're doing, one thing that's different is that we're doing a live. We have decided from now on, we are doing lives. As much as Katie doesn't want to, everything will be live now.
Yay.
She's so excited.
We got to decorate this room. This room is sad.
Yeah,
yes.
We need some anti-echo.
I'm going to work on that. I actually have a plan for a really cool quilted tablecloth.
Yeah.
That I think will help.
Here.
Yeah.
My keys.
Just so you can hear everything moving.
Yeah.
But we're also getting a new
We're
table.
also getting a different table, so that'll help.
And we'll put some posters and stuff on the walls.
Yeah, I got to decorate. This is a hot mess.
Because right now we're sitting in a room of just white walls.
And a ceiling fan.
Hey, there's Darren. We're waiting. We'll have a bunch of people, hopefully.
We got to get a ceiling fan.
Doesn't this have a green screen function?
It does have a green screen function.
What? We don't have to figure that out right now.
Yeah, I don't remember how to do it exactly.
We'll do it later.
But it's got one.
Yeah.
So I'll figure it out later.
So tell us about y'all's weekend. I was here unpacking.
We went to the farthest reaches of Texas. It was actually cool. There was one night where it was hey, look, there's Robbie. Robbie's up.
The high on Sunday in Amarillo was 74.
Yeah. I woke up cold on Saturday.
I'm so excited. Those are the temperatures that I'm supposed to have at camp next week.
That's what you're going to North Alabama for.
On a mountain.
Camp. There's Ilana has joined also. There's Victor. How's it going, Victor? I'm looking forward to having all the people on here live. It's going to be great. So, we went up to the Herp Show in Amarillo. I've never been to Amarillo. I saw a mule deer for the first time. Mm-hmm. We went herping, kind of. We went road cruising. I did find a frog or toad I've never seen in my entire life. Well, outside of pictures. I saw a spadefoot toad. Mm-hmm. These giant, yellow, lime green dots all over it. It was amazing looking.
Yeah, we started driving around after it rained.
Yeah, and then we drove around before it rained.
It was just a little too cool, I think, for any of the snakes to be out moving.
Yeah, there was a local guy that said if we had done it the week before, we'd found tons of stuff.
Like Russell,
that's cool.
Russell, Southard, and Bart, and some others, they went out, but they were flipping over a bunch of junk and found an Atrox on a what was that? I thought it was a Sonoran ground snake.
Yeah, the little stripy, ring looking thing.
That was a cool looking little snake.
So, but it was a fun trip. It was a good trip. It was the first time it's the first time Sean is in a show, a herp show there. They've had a reptile show there before. 2 years ago was the last time they had it. So I think it's going to end up being a good show. It was a decent show for us, but it was the first one there, so it's got some room to grow.
Yeah, it was okay. I mean, I did better than break even. So
yeah, you did.
I was all by myself.
Poor Katie.
But
not even my kid was here.
If I told you guys, but I heard a rumor today.
Oh Lord,
that Lone Star Reptile Racks is going out of business.
What?
And that my wife is mad.
No, there's drama, and I didn't even know about it.
I didn't know about it either.
Man,
so where did you hear all this at?
I just heard it, and somebody called to verify. They called. It was more than one person, but I know where it came from, and I've already nipped it in the bud.
That's crazy.
And told him,
hey, that's how you know you've made it when rumors fly.
Well, Todd posted a thing today on Facebook about how when he went into this full time, he had so many naysayers and stuff, and I was like, hey, I heard I'm going out of business. What you hear? He's like, yeah, that happened to me a year ago. People started saying I was going out of business.
I
didn't ask if I could share this, but I'm going to share this. My favorite was the customer that complained and gave you the BS reason for wanting his refund.
Oh yeah. Oh, you even heard the rest of that.
So what I had heard.
Okay, hold on. Wait.
Wait. What I had heard is he wanted a refund because he had heard a bunch of negative things about Lone Star Reptile Racks. Which, by the way, I've searched. There's nothing out there, and he has no negative reviews.
So I asked him, "Can you put me in contact with the people who are having a problem that you?"
Because they don't exist.
Then he said, "Well, I'll go ahead and take the rack." And I said, "No, you're good, man. I already hit the refund button. Go buy it somewhere else. I don't have time for."
Look. All right, anybody listening? If you order something that is a special order, and it's going to take time to get it to you, if at some point along the way you realize you just don't have the money for it, you need that money back, whatever. Just tell the person, hey, man, I know I ordered this, but I need that money back. And a lot of times they'll go, that's cool, and they'll refund you the money. Don't make up reasons why you don't want to get whatever it is.
Heads up, I may or may not sneeze in the next 30 seconds. I apologize in advance.
Okay.
Also, I can't talk to our friends in the chat.
Well, yeah, you can't
because
comment from
I'm
here.
Backstage.
You don't need to talk to them.
I am. Victor has a really cool picture he was going to show me.
Well, then tough. Victor can wait. Victor's like a statue. I've seen him on Zoom calls.
Uh-huh.
For half the time, I just think he's asleep.
Oh my gosh.
Or that his screen froze.
So mean. Respect your wife, James. Respect.
Victor will
your
get
wife.
on Chris's Zoom calls, and he's definitely, let's say, not at home. So I don't want to get him in trouble. He's not at home, and just frozen. It's great.
Oh my goodness.
So yes. So speaking of, let's do sponsors. Lone Star Reptile Racks not going out of business. Katie, do you want to?
In fact, if you are looking for a high quality PVC rack, look no further than Lone Star Reptile Racks. They offer a variety of sizes for all types of snakes, geckos, rats, and more. You can even order something custom. Shipping is available, or you can plan to pick up at a Herps Reptile show near you. Visit lsreptileracks.com to reach out to Lone Star Reptile Racks and place your order today.
.com. I didn't get a chance to say.
Thank God.
Also, we mentioned them, Herps Reptile Shows. Our next reptile show for Herps Reptile Shows at Slidell, which I'm sure will be crazy, as it always is. If anybody's ever been to Conroe and not been to the Herps Slidell show, they're about the same. I mean, it's the same number of people coming through that door. It's crazy. And then our others is Wiregrass Exotics, who are opening their doors officially this weekend.
Yep. T-minus 2 days until their grand opening, which will be July 3rd at 11 a.m. If you are in the area of Ozark, Alabama, be sure to go by and check them out. They'll have feeders, reptiles, and supplies. They're also going to be holding a special raffle on Saturday for lots of goodies with gift cards, supplies, and maybe even a custom-built tank. Be sure to tell your friends and family and everybody in the area that you may know who's looking for something cool to do. Dallas and Amanda are really awesome people, and they've got some really cool things going on in their shop. So be sure to check it out.
They kind of have like a feeder subscription. If I read the board right, it's $1,000, but you can get them. Or not 1,000. It's 1,000 crickets, but you can get them as needed. So like you pay for your 1,000 crickets,
and then you go in.
And then you go in. I need 100 this week.
Mm-hmm.
I need 100 next, until you reach 1,000.
Your subscription.
Mm-hmm. Yeah,
pretty smart.
Yeah, that's pretty good. That way you're not having to go buy 300 and then 150 of them die before you can ever feed.
Well, and if you're like a lot of other people and you might be living paycheck to paycheck, it's a lot easier the day after payday to say,
yes,
oh, here's $75 and I'm dropping on animal supplies. And all of a sudden, oh man, it's end of the month and I need this much, and you don't have it. But if you know you have your subscription there, you're good. So they've got their big grand opening on the 3rd. Then we have our friends over at Grant Family Exotics in Oklahoma, who also have their big grand opening of their second store. Second store? New store?
It's a new store. They moved
to a different location.
Catty corner from their old store.
Or if you live up north, it's Kitty Corner.
That's wrong. It's Catty Corner. So they moved Catty Corner from there. You can literally see the old store from their window. But they also open
this Saturday.
Yeah.
So, I mean, Oklahoma, Alabama, anywhere in between, pick a side, drive, make a trip.
Oklahoma, Alabama, or any Herp show in between.
Yeah. It's going to be, it's a great weekend for reptiles.
So, and then like I said, in 4 weeks, when is Slidell? It's the 20.
Yeah.
Slidell's the 24th and 25th of July.
What she said. 24th.
I'll be at that show. That's after camp.
Yes. That is our next big show. Looking forward to that. Although you have another show, one that we've never done before.
The shirts, Lone Star show.
Yes. Darren Watson said if he was planning on attending a Herps Reptile show, what would be the best show and what time of year? I would definitely check out either Slidell, Louisiana, or Conroe, Texas.
Yeah.
Just because I think those are the two.
And I would do the
definitely
show
largest shows.
And I would do the show somewhere around. I know there's a Conroe in September.
Yes.
That's gonna be a good one because people are gonna have a lot of
babies.
Stuff to sell babies. And I don't know when the next Slidell is. Slidell's done 3 times a year, just like Conroe. Those are the only 2 shows. Conroe and Slidell are done 3 times.
I want to say it's November.
So that would be the good one to go to for Slidell. Whichever the next one is. Not this one. I mean, some people will have some babies, but it won't be as good.
There is Slidell's December 11th and 12th.
December.
Oh, yeah.
So that's a good Christmas stuff.
But Slidell's a big one. I love that one. Plus, if you're making a trip of it, Slidell, you're 30 minutes from New Orleans.
I love everything about the Slidell show except the drive.
Yes.
Because that drive sucks.
Yes.
Well,
I'm not excited.
It has not sucked for us in the past years because we were only 3 hours away.
Because we've only been 3 hours.
But now we're in Texas and it's going to be.
Let's see. The first Slidell, the 6-hour drive took me 10.
Yeah.
Last time it took me 8. course, Lafayette took me, what does it take us?
Yeah.
5?
It's that I-10 stretch between Lake Charles.
There's always an accident on I-10.
Killed us yesterday, too.
So.
Yeah.
All right.
Brutal.
I think that's, before we go any farther, I do want to announce that I had something happen. So we were out herping, like I said, on Friday night, and I got a FaceTime call from my mother-in-law. And as soon as it hit my phone, I knew exactly what it was. I was the farthest I could possibly be from my home in Louisiana.
And still be in Texas.
And my rainbow boa had babies.
And she did call you first. I want you to know. She didn't call me first. She called you first because I was like, please tell me that you've called James. She's like, oh, I called him first. I said, good job.
And then my buddy Chuck, I had my buddy Chuck go over there at midnight. I've got to thank Chuck. He's amazing. He went over at midnight and collected all the babies. There were 10 healthy babies. They are now here in Texas with me, along with Mama, who I got to get set up tonight. There was 10 healthy babies. I think 2 stillborns, 1 very underdeveloped, and 3 infertile ovums. So not bad.
Say hi to Jennifer. She said hi.
Oh, hi, Jennifer.
Pretty awesome.
I do want to finish. I don't want people to think I'm bitching about my customers.
No, you.
Look, 99.9%
are amazing.
Customers are amazing. It's that 1%, 0.1%.
No joke.
There was the girl at Amarillo that really impressed me this weekend. She was looking at either the 6 tub, 28 quart, which was smaller, or the 3 tub 70, which would be larger, which would hold adult ball pythons their entire life. And the girl had 2 adult ball pythons, or grow out 3 adult ball pythons she was growing out. She could have gone the cheaper route and put them in the smaller one and got more cages. But instead, she made the right decision and she went with the larger one.
And we even advised her to buy the larger one, which costs less. I'm not in it just to sell everything.
Yeah.
I want you to have the right thing. But, occasionally you get, like right now we've been dealing with the PVC shortage. And I am up against the wall right now. There is none. Tomorrow's the 1st. We're supposed to get an update. Hopefully going to have some early next week. But I've got some people that are, 8 weeks right now since they ordered, and I just don't have the PVC to build, and I have to do shows.
Yeah.
That's where I make 95% of my money at.
I forgot to hit record on my little thing here.
Oh,
no, it's recording on there though. We got it recording. I just didn't record them. I'll hit it here. Maybe people will hear it spliced together later.
Fancy box.
I know.
So yeah, it's a little stressful right now, but
it's getting made, and you get a new machine soon.
Got a new machine coming in a couple days, and then I got, we made our first acrylic box today.
You can see it on the screen there.
Yeah. We made our first acrylic box today with a slide out lid, and it'd be great for isopods and
inverts stuff.
Inverts, all kinds.
I plan on doing some stuff for some small snakes, but that's down the road. So, all right, I want to go ahead and bring in our guest. I think it was Victor said, how'd the move go? The move went fine.
The move is still happening. Let's be real.
It's still happening. We made a trip yesterday to get more stuff. There's still stuff in Louisiana.
I've gotten rid of so much stuff, and there's still there's not as much to move. Like we could do 1 trailer load and get everything else that's left at the house. But then there's still a storage unit.
Yeah, we have too much stuff.
But it's cool. It'll get done when it's done.
It is. So let me make sure I bring in our guest, Dr. Hannah Seidel. Is that how you pronounce it?
Seidel. Mm-hmm.
Oh, I forgot.
You got to unmute them.
There we go. They're unmuted. Dr. Hannah Seidel.
Yep.
And Dr. Chiron Graves.
Oh, he got the first name right. I just knew you were going to mess that one up.
I cheated. And had her tell me your name.
We practiced.
I think I'm a nerd for a second. I'm pretty sure that's the name of the guy that drives the boat from the underworld in Greek mythology.
No, that's Chiron.
Okay.
Chiron is the centaur.
I was about to say he's the centaur that
I knew it was
that helps out Percy Jackson because we read that at school last year.
Definitely.
Oh,
that's what they read in 4th grade. So yeah.
Okay. Well, I had Greek mythology right. I was just wrong person. So I'll let y'all go ahead and introduce yourselves, kind of give us a little background, and then we'll get into the really interesting work that y'all are planning on doing, and y'all are doing, and kind of how everybody can help here. So whoever wants to go first, I'll let y'all decide.
I can start. So I'll first just say that I'm so happy to be here to talk about our project. So we founded what we're calling the Ball Python Genetics Project. And the goal of our project is to identify the genes and mutations that cause different ball python color morphs. So we're interested in what those genes are, what they're doing molecularly, how can we create genetic tests for them. And we have both scientific goals and educational goals because this project is part of an undergraduate lab course at Eastern Michigan University, which is where both Chiron and I are professors. We're both in the biology department. And so I teach a genetics laboratory where students learn techniques in genetics. They learn how to read scientific literature. And I wanted to create a project where the students could be collecting real data and making real discoveries in their classroom. And so the basic idea of what we do is we collect shed skins from ball pythons sent in by pet owners and breeders throughout the country. And then we try to identify the genes in different morphs. And so far we have identified the genes in 3 different morphs: the albino toffee candy morph, so those are allelic, and the lavender albino morph and the ultramel morph. And there is another group in Canada that has studied the piebald morph. So that's the totality of morphs for which collectively as scientists, we think we know the genetic basis.
So the albino ones, that's all dealing with, I don't remember how to pronounce it. Is it tyrannosine?
Tyrosinase, yeah.
Tyrosinase.
So the albino morph does not make the pigment called melanin, which is the brown to black pigment that we as humans have. We have it in our skin, we have it in our eyes, we have it in our hair. Different people have different amounts and different subtle variations on the pigment, which gives us our different hair color, skin color, eye color, and so on. And ball pythons also make this pigment, and the enzyme that makes it is called tyrosinase.
Yeah, because for anybody listening, that's when you see in boas for sure, and it's the kind of same thing with the ultramel and the albino. You'll see T positive albinos and T negative albinos, and so it's talking about
right.
The tyrosinase.
Yeah, so typically the T positive would be referring to animals that do make tyrosinase, but they have another defect that still prevents them from making melanin. Whereas the T negative animals, they don't make tyrosinase, and that's the reason why they can't make the melanin pigment.
And so the ones that are T negative, what we would normally refer to as an albino, is the way that most of us think of as an albino. It's the yellow and white snakes, or if you're talking about boas, it's the red and white snakes.
Yeah.
T positives being things like caramel albinos or ultramel or things like that still give you that kind of that tan color, but it's still got the albino look to it.
Yes, exactly. Like lavender albino in ball pythons would be an example where you get that lavender color of the skin, which is probably due to the animals making a teeny tiny amount of melanin, but much less than normal.
Gotcha.
Huh. So that's similar to, for example, people with green eyes in humans. The eye color is produced by the amount of melanin you have in your eyes. And if you don't have any melanin in your eyes, then you have blue eyes or gray eyes. And the people with that eye color, they don't have a specific blue pigment, but instead it's the way that light interacts with the physical structures in the eye. And if you have a little bit of pigment in your eye, you end up with a green eye. And if you've got more pigment, you end up with hazel or brown eyes.
And as far as, if I remember correctly, I'm also a high school biology teacher, and you're always taught early on that genetics, like if you mix a blue-eyed person with a brown-eyed person, but from my understanding, none of that really is true, and that you could pretty much have blue eyes or brown eyes pop up, not randomly, but not in a simple genetic way.
That's a really good question. So eye color is, the answer is more complicated than you think. It's both simple and complicated. So the simple part is that most of the distinction between light colored eyes, and by light colored, I mean blue, gray, really light green that sometimes looks blue, all of that is the difference between those eye colors and everything else, which would be hazel, light brown, dark brown, combinations of stuff, that is mostly controlled by one gene. But once you get into either of those groups, so what determines like gray versus bright blue, or what determines hazel versus brown, then the genetics is really complicated.
All right. So we have one listener, and a friend of ours, Travis Wyman, who is a professor at Travis. I don't want to mess it up. You're somewhere in Virginia. I should probably know this, but I don't.
Well, we know it's Virginia.
We do know it's Virginia.
We do know it's Virginia.
As much as Katie told me it was West Virginia.
Because I said West Virginia and was wrong.
But so he said blue-eyed people still have melanin. A true albinistic human has red eyes.
So he's right. So I simplified a little bit. So it has to do with where in the eye. So in your eye, a brown-eyed person has melanin in the retina and the iris. A blue-eyed person has melanin in the retina, but not the iris. Whereas an albino person would lack melanin in both of those places.
Yeah. So Travis, cool your ass off. It's fine. Chill.
Well, I'm glad that he brought that up. It's actually a really good point, because one of the issues that people with albinism have is they have vision defects, and that's due to the defects in the retina. Whereas people with blue eyes don't have vision defects. Because their retina is just fine.
Gotcha. And I gotta correct. I'm sorry, Travis. Dr. Wyman. My bad. I called you a professor. It's Dr. Wyman. I get it. You went to school longer than the rest of us. Special ass.
And he still bakes better than you do.
Anyways.
So he bakes, so that's automatically better than
So, Chiron, I understand you do have Hannah doesn't have snakes. She's genetics, but Chiron, you also do own snakes, correct?
I do own. I've got a small collection, but yes, I do own ball pythons, and I actually have an albino corn snake as well.
What kind of?
You mentioned that you teach high school biology. One of the things I do at Eastern is I teach teachers how to teach science. So
that's awesome.
So we're coming at this project from both angles, both the traditional science angle, but then there's that educational angle that Hannah talked about, where we're getting students involved. And I think that's one of the cool things about our project, is that we're getting students involved in this process of learning about how science is done, and at the same time, learning a little bit about pythons, ball pythons in particular, which, as you all know, you can kind of get a mixed bag with some people that are just totally, oh no, python, no. Those are the ones that eat babies, and others that are just kind of like, sign me up. I want to hold one.
Yes.
We get that. I've had some classes. I've been doing it for 4 years now, 4 or 5 years, where I'll bring some of mine in, and particularly one of the albinos and one of the wild type, to kind of talk about those differences in pigmentation and stuff. And invariably you get some students, they're like, could you have given us a heads up so we could have missed today? And then others who are like, this is the greatest class ever. It only goes down here from here, Graves. And so it's pretty fun.
It amazes me whenever James and I, because I'm an elementary school teacher and he's a high school teacher. And so in Louisiana, before we moved, we would actually take some of our reptiles to the summer school programs. And so the kids could see them up close and do educational programs with them, because neither one of us taught summer school. But it never failed when it got to the point where the snake would come out. There would always be teachers that were like, deuces, and they would like go out the door, and then they'd like wait for us to finish, and then they'd come back in the room with their kids. And I'm like, y'all are missing the best part.
I bring in some of my corn snakes when I do genetics. I bring in an anery, an albino, and a snow. I'll try to bring in a normal-ish one, and we can do some Punnett squares, some simple Punnett squares, and biology with corn snakes, just to give them a visual. Like, this is albino, this is an anery, and if I mix them together, it comes out looking normal. It looks like this. I can literally show them what it would look like, and then if you breed it again, then it comes out this color. So I enjoy bringing snakes in. I also bring in reptiles when I do classification. We'll do a tortoise, a lizard, and a snake, and do kind of a Venn diagram and similarities and differences. But for a lot of my kids, I taught at inner city school before I moved here, and they've never seen any of those animals. They knew dog, cat, and squirrel, and past that, they have no idea that it exists. So being able to bring those animals in was always a ton of fun. And it's always a lot of fun when it scares the adults at the school way more than it scares the kids.
Oh, yeah.
My administration would never come into my room when they knew I had reptiles.
We were known as he was the snake guy and I was the snake guy's wife throughout the district. Like other teachers would see me and they're like, oh, you're the snake guy's wife. And I'm like, I have my own job and career and do a lot with curriculum, but yes, that would be me. Yeah. Forget my attributes that I have coming, but it's all about the snake.
So Victor asked real quick, he said he wonders if animals with albinism also have visual defects, and I'm pretty sure they do, but I'll let y'all kind of expand on that.
Probably yes, especially the animals that are tyrosinase negative and have essentially no tyrosinase. Probably that's the case, although snakes rely much less on vision than mammals do. So a snake who is blind is probably going to have less of a hard time in life than a human who's blind, because a snake uses other senses to, for example, find its food.
Gotcha. Yeah, and I knew that there were issues. I have found that my snow corn quickly grew like cataracts in her eyes, whereas my other corn snakes didn't really.
Oh yeah, that's really interesting that you say that. There's certain genes that that cause that. So that particular detail is something that as a geneticist I would like make a note about when thinking about what could be the genetic cause for that.
So, Darren asked, as a teacher, do you need permission to bring in reptiles? Yeah, kind of.
It depends on the district. On the college level, I have no idea. On the elementary and
at the college level, you can do whatever you.
And
I think we can do whatever we want.
Yeah.
I don't know. What do you think, Kyron?
If we were supposed to give permission,
I always give my administration a heads up, and I've never really had anybody say no. So
yeah, but K-12 has always been, because I actually used to teach high school and middle school as well, and I'm originally from New Orleans, so I used to teach in NOPS.
Yeah, we were in Central Louisiana. We were in Alexandria.
What's that?
We were in Central Louisiana in Alexandria.
Okay.
Up until a month ago.
Yep.
And now we're in Texas.
2 weeks ago.
Yeah, I'd say 2 weeks. Yeah. Now
I don't know that we've been here a full month yet.
So not enough time to really get your feet wet and figure out if you like the change.
No.
It's a lot of change all at
Still
once.
unpacking.
That'll be fun.
Yeah.
So are y'all, I didn't hear you're doing this for undergrad, which I think is amazing, because I wish that was my undergrad. I could have done that. I hated my genetics. I didn't hate my genetics. I hated one part of my genetics class, and that was the damn fruit flies.
Oh yeah, I did fruit flies in my genetics class as well.
I despise them. I could not keep them alive. I kept getting mold growing in every culture. I ended up just cheating off of my buddy who had the same mutation as me and tweaked some numbers.
Sure.
But it was a nightmare. It's the reason I don't own as much as I'd like. I'd love to have dart frogs, but in order to have dart frogs, you have to raise fruit flies. And I can't do it. I have like PTSD from fruit flies in genetics class.
No joke.
Yeah, so in the criteria for coming up with this project, the first two criteria were interesting to the students and interesting to the instructor. And also on that list was something that was like methodologically easy for the students. And snakes are pretty easy in the sense that because we're just getting the DNA out of the shed skin, I don't have to be growing fruit flies in a room to get them ready for the students. The sheds can just sit in my freezer. They can sit in my office. It's really flexible, and it's a sense, it's methodologically much easier.
So what is, if you can give us kind of a dumbed down version of how is this working? You're taking a shed from somebody with, I'm assuming they're giving you a shed from a known visual of that morph?
Yeah, so yes, exactly. So I've got a database. I have almost 400 sheds now, but that shouldn't make you think that I have enough. I still want more. So if anybody's listening and they have ball pythons, please, I want sheds from all of your animals. So people will shed in a shed. They will tell me what they know about it. So they'll say something like normal, might be het for pied, or pastel, possibly leopard, something like that. Or sometimes they'll say, somebody gave me the steak, I don't really know what it is, and here's a photograph of it. And then we have this collection of sheds. And then when we decide we're going to analyze a certain morph, we get sheds out of our freezer belonging to that morph. And then we compare that DNA to normal animals or to animals of different morphs.
I'm so sorry about that.
I guess Sean Gray messaged me. I was trying to message him. Y'all continue the conversation.
I'm a very loud sneezer. I'm so sorry.
Is that the only thing you're loud about?
Okay, shut it. Nobody asked.
And I would say actually the hardest part about the whole project is actually just getting people to send us sheds from their animals.
Well,
that's surprisingly more difficult than you think.
We can fix that. I promise you.
I wonder why.
Well, it's funny because
she said the hardest thing is getting them to send her sheds.
Which I laugh because Dr. Why can I think? I keep wanting to say Wyman, but not Wyman. Warren Booth did a lot of work with parthenogenesis, and at this point, he's received so many sheds. He's like, stop. He can't take any. He knows it happens. He's done. But we can definitely get you more sheds, and we will share that information for sure. I guess one question I have is, when you're looking at that, how do you handle, because I don't know, Hannah, if you're familiar, but there's no such thing as a normal ball python anymore. It doesn't exist. Every single ball python has 15 million hets in it.
Yep. So
And
yeah. So how do we deal with like the variation that you see? Yeah. So that's why we've started with morphs that were really obvious, in the sense of albino versus not albino is a pretty big difference. And so we started with morphs where there isn't a lot of controversy about whether the animal is or isn't this morph. I will say that even within the albino toffee candy, we took an approach where we thought, people might not be completely right about what morph they are. And we found that was true in a sense of sometimes people would say that their animal was toffino, which is one allele albino, one allele toffee. And when we actually did the genetics, we found that it wasn't. It was actually just albino, or sometimes vice versa. So we do take into consideration that a person's information about their morph might not be 100% correct. And moving forward, we still plan to work with morphs where the actual trait is much more obvious to identify than some of the more subtle ones.
So Darren had a question. I don't know if this ties into... Go ahead. I'm going to be honest. I'm only comprehending and understanding maybe a third of this conversation. And that's just because, I teach. No. Anyways.
I'm good. I'm pretty good. Katie has almost no science background, and she doesn't keep snakes. So she's here for moral support.
I am. I'm the layman's term individual.
Robert has some snakes, but he doesn't have the science background.
I am understanding a little bit every now and then.
Let me know when you don't understand something, because that's really, I think, my failure to explain it in a way that someone without a PhD in genetics can understand. I don't think you should feel like that's your failure not to understand it. It's my failure not to explain it correctly.
So Darren asked, how are both doctors how are they identifying?
So how are y'all at identifying morphs? I know Chiron has some background because he has some of them. How good are y'all at just visually being able, can you visually do it? And then when you look at the DNA, you're able to go, oh yeah, that's definitely what's in there. I can see that's there.
So most of what we've been working with, like Hannah said, it's not debatable. I mean, like the difference between an albino and whether it be wild type or inchy or yellow belly or whatever, if it's albino, it's albino.
Yeah.
So that's indisputable. And so a lot of what we've worked with, one of the reasons why we work with it, I would actually add that there are two reasons why we chose what we chose. One is because this is an early project. It's undergrads. We want to make sure we can kind of get this process right. And so we want to make sure there is, without a doubt, you can look at it and tell there's definitely, this is a albino morph versus this is a wild type. The other piece is that with albinism, we know a lot about albinism because of all the work that scientists have done in other animals, in humans. So I think one of the cool things about this project is being able to say, hey, that albino that you have, that albino actually has a similar mutation that humans have, human albinos have. And that's kind of like that, James, you were talking about teaching specifically genetics, but then you could bring the evolution piece in there and how we're related, in the sense that, wait, snakes are using the same gene that humans use to make melanin. So, but most of what we use, most of what we've been doing so far has been, it's undisputable. And as we get the procedures down, and we're really comfortable with that methodology, then we'll start going into things where it's like people have a difficulty spotting it, like yellow belly, right?
Yeah.
And so that's the goal.
So our buddy Sean, Sean Gray, he used to breed leopard geckos, and he tried before and I talked to him this weekend, reaching out to some companies. I think it was in Japan. He was trying to do some genetics work back then to try and figure out certain things. And he had a question. His question was, how are you at handling the issue with incompatible alleles regarding different albino strains? And is this just for homozygous traits?
Yeah, so good question. So in ball pythons, there are a bunch of morphs that to a geneticist would be some version of albino. So earlier you talked about tyrosinase negative and tyrosinase positive. There are a bunch of genes that make a bunch of different proteins that are required for making the pigment. If you mess up any one of those genes, you're going to get an animal that looks albino. depending on the gene you mess up, you might get a animal that is more bright white than a different animal. And so those would be different genes. So I think when he's saying, how are you handling the issues with incompatible alleles regarding different albino strains? I think he's referring to what we would call different morphs, like the albino morph versus the lavender albino morph versus the ultramel morph. So we treated those different morphs differently. So we said, let's look just in lavender albinos, and let's look just in ultramels, and let's look just in albinos to, so I guess the way we handled them is we treated them as different groups.
And so he said, can you differentiate the genetic markers between the albino strains? So could you differentiate lavender albino from a regular, what we call a regular albino?
Yes. Yeah. So let's say somebody received a snake and they didn't know what it was, but they knew it looked like it was either albino or lavender albino or toffee or some version of that. If they didn't know actually the genetics of it, they could send us a shed and then we could do a test and we could tell them exactly which one it was.
Oh, wow.
And let me just jump in real quick, because this goes back to the piece that Hannah was talking about with trying to get people to send us sheds. So it benefits both the breeders and us as well. So as educators, we're trying to find that real connection where we don't just have to, like something relevant, right? By the students working to find an answer to whether or not my hatchling is het toffee or het albino, because I bred it to a toffino, and so it's got to have one of those alleles, but I don't know which one it is. It benefits the breeder because now they could, instead of saying either het albino or het toffee, be able to have that definitive, it's het albino or it's het toffee, and not have to put the either or in there. And it benefits us because we get those samples and we've got a goal for our students to shoot for. Hey, this breeder, and if we could report who the breeders are and they're what they're wanting to know, they want an answer for what this genotype is. And so we can kind of help them out with this process.
Yeah, so one of the projects we have going for the fall is we are trying to recruit samples from breeders or owners where the owner or the breeder would benefit from knowing more about the genetics of their animal. So if someone has an animal that they think might be possibly heterozygous for any of the morphs I just mentioned, lavender albino, ultramel, albino, toffee, candy, if they send us a shed, we can tell them whether their animal is or is not het for that morph.
Gotcha. And so I was looking, our buddy Sean, to clarify, because I remember he told us about this. His worry was, how can you differentiate the markers from a het and a non-heterous one? So like, it's so hard nowadays to get a clean sample of an animal that doesn't have any hets. It's very hard to find a lot of like corn snakes. You pretty much guarantee like they're het albino or het anarine animals. Any corn snake, how could you promise in a ball python? How do you know when you're breaking down the markers that you got a clean sample of one that has no hets in it, so you can compare the rest of them to that?
So I think what you're asking is, let's say that I received a shed from someone who told me that their animal was a normal morph, that it wasn't het for anything. Yes. You're saying that in theory, it's possible that animal could be het for albino, but that person wouldn't have known it because you can't see the albino trait when it's het. Is that what you're asking?
Yes, because just it's near impossible nowadays to find a non-heterozygous animal out there.
So that's a really good question, and we have a genetic test that allows us to tell at specifically a particular site in the genome whether the animal has or does not have a particular mutation. And once we've identified that mutation, then we can distinguish animals that are not het to animals that are het to animals that are homozygous. And when we did our initial tests, we collected 45 animals that were, for example, for the albino trait, were not albino. So the people told us that their animals were not albino. In that sample of 45 animals, we ended up finding one that was actually het for albino, but the owner didn't know about it. And we were able to identify that with our test.
Gotcha. So I don't know if you realize this. If you guys are able to do this across basically all the morphs eventually, as far as the hobby goes, it'll be a huge thing because there's so many snakes sold as possible hets, probable hets, and that gets very confusing.
And
there's so many
that's
snakes.
why I'm surprised more people haven't sent in their sheds to me.
And now we got another geneticist on here. So crap, now I got doctors everywhere. Let's see. Let me read Warren's. How many mutations have been identified to the genomic region? I assume it's simply through RADSEQ or WGS to identify common markers.
Yes. Okay, so he's referring to two different techniques. The first one is RADSEQ. The second one, the WGS stands for whole genome sequencing, I believe. And the answer is that in ball pythons, the morphs whose mutations have been identified are albino, toffee and candy, lavender albino, ultramel, and piebald. That's for ball pythons. There is a study in leopard geckos that has found the mutation for, I think it's called the frosty morph. It's a morph that has extra white in its body. And our study did not use either of the techniques that he mentioned, but the study on piebald did use those techniques, and so did the leopard gecko study.
Gotcha.
And the plan is moving forward that we're going to do additional morphs. We're going to move on to more of them.
Yeah, you're just trying to get a proof of concept now and move from there.
Yes, and that's why we started with what to our eyes was essentially the simplest morphs, which were the albino, lavender albino, ultramel morphs.
Okay, you're going to have to read those and let me know if it's important in a second.
Oh, no, it is. And I was actually just about to interrupt you. Dr. Booth said that leopard gecko, it was lemon frost.
Oh, yes. Thank you.
The lemon frost. Yeah. So, Hannah, we wanted to make sure that we weren't misrepresenting anything. Dr. Wyman's speculating just a little bit.
Talk to the mic.
Basically, I was scratching my nose and did not want to do that in the microphone. Thank you so much. I moved away to scratch my nose. Basically, you run a test out on a gel and the WT gene leaves a band at a certain distance. Yeah. The albino gene band runs at a different distance. Yeah. So a homozygous, you would only see one band, but in hets, you're going to see two bands because of the two different genes present.
Yes, what I'm trying to find for you right now, I have my lab notebook in front of me. I'm trying to find an example of a test I did for someone that's actually
this is what happens when we get way too many doctors at one time.
For lavender albino, and it shows it really clearly, but I can't find it at the moment. I think it's in a different lab notebook.
So it is very possible that we have already answered this question, and
you were confused the first time.
And why is this important?
Well, it depends on which way you're talking.
No.
To them
like
or to
well. I mean, that's why is this important to Dr. Graves? Why is this important to Dr. Hannah? Like, why is this important to you? It's not just your work. Like, otherwise you would be, doing something else. But what makes this what you pick?
Yeah. So I would say that it's a variety, and it's important in a variety of different ways. So one is that we find that the students are more engaged in class when they feel like when they know that they're actually making a discovery. So we literally, before this project, didn't know which mutations cause these morphs, and now we do because of the data that our students collected.
That's cool.
That makes them more motivated to learn about the literature, to learn about the techniques. They're excited about the project. So it's an educational goal in a sense.
Yeah.
And another goal is simply that there are ball python breeders who would like to know this information because it would allow them to make morph combinations more easily if they could know for sure that their animal was
this, and this would give this. Absolutely. That's pretty cool.
In that last point, Hannah mentioned, it's like both of those are important points for me. One, because I'm a science educator, so I'm looking for ways to teach my future teachers how to, teach in a way that's more engaging. But the second part is I own ball pythons. I have a tall pheno. It would be very advantageous to me to know that the hets are either het albino or het toffee, and we've got a test that can tell me that.
So that way you can breed if you want.
Right. I can sell it as het toffee, or if the test tells me the allele that they got was the albino allele, I can sell it as het albino, and that would be very satisfying for me as a breeder. But I'm sure my customers would also, kind of benefit from knowing, okay, this is definitely a het albino, and I can kind of move forward with that.
That's cool. And like I said, that very well could have been brought up earlier. And I just with everything at once, it didn't click, but that's really cool. So I
would also just add one more thing, if you don't mind, that in fact, so there's like a third reason, which is just that science doesn't know. We as a collective group of scientists don't understand much about the genes that control coloration in reptiles. We know about humans we know about mice and humans because people study them, but basically nobody is analyzing the genetics of reptiles. And the reason is that reptiles are hard to grow in the lab. They have long lifespans.
That makes sense.
And essentially the ball pythons, because people will pay more for an interesting looking animal, essentially the United States is full of animals that are like a resource for geneticists to learn about the coloration of reptiles that we wouldn't otherwise know about.
Yeah, it's a country full of lab specimens.
Yeah, no.
Yeah, so I'm like,
that's cool
to use that word, because so to me, when I see an albino, I see a mutant, but I know to an owner, they're not thinking of their animal as a mutant. So I don't want to say that the United States is full of mutant animals, but
so it's like my husband. It's like my husband, who through my entire pregnancy referred to her as a parasite.
She was a parasite
because of the definition of parasite.
Babies are parasites. Well, fetuses are parasites.
The whole time, people would look at him so weird when he would do something.
Feeds off the host and dies on its own.
Oh my gosh. So to go back through the chat, Travis, you're not being annoying. I mean, I'm sure that they are thoroughly enjoying the conversation with people that are definitely on their level.
I have a question, Hannah. I know why Chiron is interested in this. I can't imagine prior to this you went, ooh, I really want to study snakes.
You're sort of right. Although now that I'm in it, they're really interesting. So before this, I did not study snakes. I actually studied worms, which are a similar body plan, just much smaller. And the reason that I got interested in this was because when I first taught my genetics lab, I thought, this is boring. This is boring for the students because they're not discovering anything new. This is boring for me as the instructor because I know what the outcome to all these experiments is going to be. This is boring. I want my life to be more interesting. And the way I'm going to do that is I'm going to come up with a project where my students can actually collect new data that we can publish, that the students will be excited about and that I will be excited about.
That's awesome.
And then I went to have a drink with Chiron and he said to me, I have these snakes. And I was like, tell me more about them. And that's how it got started.
That's cool.
All great ideas start from over a beer, right?
I mean, yeah, we live in the South. We totally get that.
Well, I imagine, Chiron, I really got into corn snakes at one point just because I could play the genetics game and they were much cheaper than ball pythons. I could mix this and that. And I love working a Punnett square because I'm a weird nerd and Punnett squares are fun. So I totally get you going, well, I've got these animals. This would totally fit in with what we're doing and going that way.
Yeah.
Now, I assume the great thing about this is right now it's an undergrad program, but I'm assuming long term you could turn it into some sort of graduate program or turn it into something more. Because I'm just thinking that if you expand this is technically a business idea. It's a great way to raise money for a university or for anything like that, along with also doing research.
Yeah. So I may eventually, if we do a lot of genetic testing, need to charge people for the cost of supplies, which would be not a very big fee. It would be like $5, $10, $15 or something like that.
People will pay that, no problem.
But I don't plan to start a company and make money. So, and all of our results are public, like we're going to make them public. So if somebody wanted to start a testing service, as a scientist, I view my job as learning about the world and telling people about it. So you don't expect to see me CEO of ballpythongenetictesting.com. But I do hope to expand the project in the sense of working with more morphs and getting more students involved, potentially outside of the class I teach.
That's awesome. A couple of comments from our listeners. Victor laughed and said that we've opened Pandora's box with all these questions and all the different things, but he said it's making very interesting conversation.
Well, and they started with the one species that would definitely get the most interest, being a ball python. I mean, that's the number one in the hobby, especially as far as morphs, and it's definitely going to be. at this point, have you had people reach out to you and basically say that what you're doing isn't possible? Because I just assume with something like this, people will play it down sometimes and say, it's probably not going to happen.
I have not had that. What I've had is people asking me to do things that are really outside of this. Like someone will say, oh, that's so cool that you're doing ball pythons. I have a dog that might be a German Shepherd mix, but I'm not sure. Can I send you a sample? Or they'll say, I have, I forgot what somebody emailed me the other day, a certain, like some type of salamander, and they wanted a sex determination test on it. And that's an interesting question, but I don't have the resources to also be working on that.
That's funny because Warren Booth said sex determination was determined over mini beers. So
yeah. And Sean Gray posted that he was driving earlier, which is why he wasn't really that involved in the chat, but that things are making a lot more sense now after hearing you, because he's listening to this live in his car. So he said things are making a lot more sense. He had a couple of other questions though.
I recognize who Warren Booth was. He's the guy who had the paper about the XY in boas and pythons. Am I correct on that?
Sean did leopard geckos.
No, Sean just bred.
Yeah.
He's not a doctor. Warren,
very knowledgeable. Not that there is anything against not being or being a doctor.
But there is
Warren Booth. Am I?
Yeah, that's definitely.
Yeah, that's Warren Booth.
Warren does work with boas.
Okay.
I'm trying to catch up.
That's a good question, Sean.
Yes, so Warren is right. Everything is possible. It comes down to cost. That's right.
Yeah.
Yeah, and Warren said yes. That was correct. That is.
Somebody said, are there limitations as to what genes could be identified? And I would say that a limitation is that. So I don't know if this will be controversial if I say this, that there are some things that ball python breeders or owners call a distinct morph that to me as a geneticist looks like that the genetics are more complex than just a single gene there, where there's a lot of variation both within what animals are calling the morph and not. And I think those are ones where determining which animals were that morph or not would be a difficulty, a challenge in the project.
Yeah, I would love to be able to say yes, desert ghost, and I've already forgotten.
Yeah, so desert ghost is one of the ones that comes to mind that I feel like there's quite a bit of variation and
yeah.
So.
Well, and it's weird. So in the beginning of all this genetic stuff with ball pythons, they were coming in on like 1 or 2 shipments from Africa and then getting spread across the country and then bred, and someone in California, New York, and Florida all had the same thing, and they called it 3 different things. It's gotten
yes, exactly.
So it's gotten a little better now, but there's still a lot of, if I breed that thing over there, you're saying is one, and I breed this over here, you're saying is different. When I breed them together, I get one. So it obviously can't be different.
That's right.
If I'm getting one.
Yeah. So when we do our genetic analyses, we typically lump together things that when you breed them together, you get the same thing. So, and that helps us with our genetics.
Yeah.
Because you were talking about the lines and the markers. And so in my mind, it's like a pregnancy stick.
Well, it's a gel.
Obviously, that's not what it looks like. But in my mind, that's what I visualized.
You're not
James is over here like, I'm going to divorce you after
No, you're not wrong. You're wrong.
You're not wrong. So that's what's called, I think a pregnancy test is typically some type of laminar flow assay, and you're basically flowing things over something. And that's I'm surprised how much of biology is just like moving things through something. And the test that we do is really similar. So we essentially we create what would look to you like a piece of Jell-O. It's made out of what is sold in the grocery store as vegetarian Jell-O, and then we collect DNA from the animals, and we do a biochemical reaction that basically makes more of a particular region of the genome. So we get a lot of DNA, but only corresponding to one particular gene.
So you're using PCR, I'm assuming?
Yes, we're using PCR, yes. And then,
I know what that is.
Yep. So we force that stuff to move through this Jell-O-like substance, and depending on how big the DNA is, it can either move fast or it can move slow, because if it's bigger, it just can't move as quickly. And then we look at where it is, just like a pregnancy test. And it gives us information about the genome, about the genetics.
So you had to have seen this in like basic biology. It's like a blue gel.
Do you know how long ago that was?
Yeah, but it's a blue gel with lines on it. You don't remember those pictures at all?
No.
Oh, okay. Well,
I made an A in the advanced class, and then I moved on because I knew I didn't need it for my degree.
Y'all didn't do like electrogeophoresis and
I don't even know what that is.
you hook up to like.
Nope, that was all you.
Can you guys see that?
Yes.
So it's a bunch of lines. It doesn't look exactly like a pregnancy test, but you get the idea that we're seeing black stuff or not
Right.
black stuff.
So that was my next question. Like James was saying, you have a guy in California and Florida and in New York, and they all have possibly the same genetic morph, but they're calling it different things. Do those morphs look the same on your test so that you can say, oh, we know that this is this, even though it's three different things, we know it's really the same thing.
Right. So we don't know until we analyze the morph, and the answer is sometimes yes, sometimes no. So let me give you some examples. Toffee and candy are the same genetically.
Okay.
Albino, there's actually 2 different alleles for albino, but breeders and owners didn't know that. And so when we do the genetics for albino, we have to look for 2 of those because one or the other or a combination of them all gives the albino trait.
That's really cool. But yeah, I mean, Sean definitely said this could absolutely change our entire hobby. And
for the albino, those 2 alleles, same spot on the DNA or 2 different locations and have to happen at the same time?
So they're in the same gene, but they're in different locations in the gene. They're actually pretty close together. So they're like 30 base pairs apart. So it would be like if the gene is a sentence, one of them is in the 3rd word of the sentence and the other one's in the 5th word of the sentence would be an analogy.
Yeah, that's another thing you'll find talking with snake people and non-science people is that you have people that barely passed high school biology trying to explain genetics to people, and it gets very confusing when you start throwing in incomplete dominance and recessive and dominant, and so a lot of that gets really confusing as well for the general public because they're hearing these words for the first time. I was trying to read what Warren said. They can be different mutations of the same gene through independent evolutionary paths. Same genes and likely different alleles of the same genes.
Yeah. And hence compatible.
And hence they're compatible. Yes. And Travis said he was surprised there's only 2 for albino. Sorry to let you down.
That we know of so far.
Right.
Yeah. So we had a total of 27 animals in our study that were either albino, toffee, candy, or some combination of those. It's in theory possible that if we collected another 20, 30 samples, we might find an additional mutation somewhere, but so far it looks like it's just those.
It's crazy. I think of this being used for hets is going to change how people sell stuff from now on, especially because you can go. The crazy thing is if someone goes to PetSmart and buys a snake, right? They go buy a ball python from PetSmart and they label it a fancy ball python. It's whatever color and they have no clue. You can take that shed, send to you, and go, oh, this fancy ball python has this, this, and this in it.
Right.
These markers.
I only paid $300 for it.
Well, there'd be no more pos hets.
Yeah.
I mean, or no more 66% hets. It would be, here's the paper showing what the hets are,
which is good because those numbers really confuse the general public. When you tell somebody it's a 66% head or a 50% head, they don't understand what that means. And when you're trying, it's chance. It's a 50% chance of it not be. It's not like half of it is het and half of it's not het. That's not how that works.
Yeah, that took me a little bit to grasp when we first started dating. I was like, I don't understand. It's not half.
The left hemipene is het and the right hemipene is not. You got to hope it breeds with the right one.
Like the big argument right now is that stranger and I think cypress are the same thing. And because people are saying if you breed a stranger to a stranger, you can get a summa, which proves out that they're cypress. And this, if this got advanced to that point where you could prove that that's the same gene. One's an inexpensive gene and one's a really expensive gene.
So here's my question.
It would definitely
when we're talking allelic genes.
Yeah.
Because there's many things in the Bell complex, the blue-eyed leucistic complex gives you a white snake. And there's many different things that obviously look different. So Lesser and Mojave definitely look different. Can y'all see that in a genetic test? These 3 things that are all allelic and all end up giving you the same thing. Can you see those things on your genetic test? They are different from each other.
So the answer there's a little bit complicated. So we haven't analyzed that particular complex of morphs, so I can't say for sure. But what we see as a geneticist, the way I would describe that morph is that there's variation in the morph. So even in like a super Mojave, or if you mix a Mojave with a lesser, you get there's some variability in what the animal actually looks like. that variability could come from two different sources. It could come from the actual allele itself, or it could come from other pieces in the genome that are not that gene, but are other things that are contributing to the phenotype. And
just like polygenic differences.
Yes, exactly. So it could be that those are all the same allele in one gene, but there's other differences on the same chromosome or elsewhere in the genome that are contributing to those differences. And in that case, we would know, for example, that the animal was one of those alleles, but we wouldn't necessarily be able to tell exactly what it would end up looking like. The other possibility is that they are different alleles of the same gene. In that case, we would be able to make a better prediction. I recognize that's not a very simple answer, but I guess the summary is it might be more complicated than you think.
That's the whole thing about genetics. None of it is, as much as we like to simplify, like this is albino, this is not albino. That's the very simplified version, but there's a lot going on there. And it's not always, like you said with albino, it's not always just one spot on the DNA that changes that. Right. So it's a complex thing.
It's at least up to 2 spots now. It could possibly be more.
Albino, there's at least 2
For
spots.
now, there's at least 2. So Victor said that he always thought the 66% was a greater chance to hit the odds.
Yeah, it's a 66% chance.
I will admit, like yes, we have several different types of snakes in our house. And yes, I could take care of them if I needed to. And I could tell you a little bit about each one of them, not genetically, but just about the species as a whole. But I always get so excited whenever like a litter of Samboas are born, because I'm like, all right, what kind of babies are we having in this litter? Because we don't ever know what's going to come.
Well, no. You don't know.
I don't ever know.
I know.
James
I don't know
knows what's coming out of the litter. So, we'll have normals and we'll have aneries and things like that. So by knowing the traits, obviously it doesn't guarantee that you'll have this offspring.
Yeah. So what you're saying is if we had this test, it would make it less exciting for you but on one hand, for the people that want to know specifically what they're breeding for specific breeding projects, it would make everything better.
Well, it's still an odds game. I mean, you're still talking odds when they breed. It's a 50% chance this will happen for this one baby, and then a 50% chance it'll happen again for this one, and so on and so forth. So I mean, it's still, but this would definitely clarify. There's no more of like, all right, we'll only breed these 2 things together and see if it's got this gene. Because I've got a few snakes like that I'm raising up, and then in 2 years, when I breed them together and an albino doesn't pop out, I'm gonna be kind of heartbroken a little bit, because I mean I knew there was a chance it was het albino, but it would be nice to be able to go, okay, it definitely is.
So Dr. Booth said in that case, simple breeding experiments with unrelated females would determine that through breaking breeding experiments. So I'm not sure if that had to do with us.
Melts my brain sometimes.
Or what was going on. That may have been what was going on in the chat. That may have had nothing to do with our conversation. But on a better topic, Sean Gray wants to know if there's a place where he can make donations to help with this research.
Yeah, so he's asked me that. And, scientists are not used to receiving donations. So I have asked the head of my department if I'm able to receive donations. And the answer is yes, but I need to figure out the logistics of that.
Gotcha.
There you go, Sean.
Well, when you know, let us know, and we'll share and post the information.
Darren's question. As a scientist, do the common names bother you? Do you tend, when you're in the lab, are you referring to these by some sort of more scientific name based on just the gene itself, like gene X or gene whatever? Or are you using our ridiculous names we use in the hobby?
I'm using the science names. And it was actually really hard for me to learn the logic of the morph names. For example, yellow belly versus ivory, how ivory is the super yellow belly, and also even just the word super.
The super instead of homozygous.
Yeah, it took me a long time to understand what was going on there. But now I guess I feel pretty comfortable with it. And I would say if I were to change one thing about the naming of morphs, it would be what we talked about earlier, how there's some morphs that have a bunch of different names because they were found by different people and they're actually the same thing.
Yes.
Yeah, absolutely.
Trust me, that drives people in the hobby nuts also.
So Warren Booth said that was in relation to the gene complexes. It would be breaking gene complexes.
Oh, yeah. Okay. Yes, he's right. Yes.
He also said that you could set up a gift account
for the
donations.
Okay.
So I make a note of that.
Said your accounts department can set that up easily. They may expect some level of overhead, however.
I'm just trying to get them to respond to my emails.
I can see where that would be hard. Yes.
She's not to that step yet, but now you know what steps. He said at that point, it's similar to any grant that you would receive.
Yeah.
So that'll definitely help out.
I will say with some of the morphs, so one of the morphs we studied was ultramel. And there are some other morphs that look like they are kind of similar to things that reduce the pigment melanin, the black to brown coloration. And so those examples are Monarch and Rainbow are some examples. And I've been trying to collect samples of those, but some breeders have told me that they're hesitant to send me a sample because if I find genetically that it's actually just an ultramel, then their animals will be worth less.
Yeah.
And so that's been a challenge for is that breeders in some contexts may not actually want to learn the genetics.
Yeah, that's the one problem I was thinking, because you're gonna find lots of breeders, but this will be a big thing for them.
I'm not saying that those are ultramel. I actually, there's several other genes that they certainly could be. So if you are listening and you have a Rainbow or a Monarch, definitely I would like to have samples, but that is a challenge for me.
I do know somebody with a lot of Monarchs, so maybe I can reach out to her and get some stuff sent to you with some Monarch DNA, because she's got like 100 of them. So let me ask you, Chiron, what do you plan on doing? Is this just a small hobby for you, or you want to get more snakes and breed more and do more? Kind of moving away from the snake stuff.
Right now my collection stands at 22.
Not bad.
That's not a pretty good little size.
It's all relative, right? Yes, my wife. Like, he's got way too many. He's got 21 more than he should have. But my goal is to I'm just doing it as a hobby. And I just like the way some of the morphs look. In particular, I've grown a pretty strong affinity for clown and for the albino candy toffee complex. And I've got a project where I would love to combine those two into, the double recessive in that regard. But in terms of like expanding beyond the numbers that I have, I want to keep it down to like maybe 20. Because the thing is, it's easy for me to kind of maintain that as a hobby and just, kind of a break from my normal day-to-day activity. I mean, I come in my snake room. I'm in my snake room now. It's kind of getting dark because the sun's going down. But I come in my snake room as a kind of a de-stressor.
Yeah.
If I go beyond, if I get too many snakes beyond 20, all of a sudden the dynamic changes.
Now you have another job.
More time with them, Yeah. And so it is a hobby for me. But at the same time, it's also kind of looking for angles to provide experiences for my students to kind of learn genetics in a way that, like Hannah and I have been saying the whole time, isn't as boring as the way we learned it.
Yeah. Have you bred anything yet? Have you reproduced anything, or are you still in the grow up phase with all your snakes?
So actually, some of the data that is in our paper comes from some of the pairings that I've done with. So I've got one in particular that I'm thinking about is Axanthic VPI that's het for albino. And I crossed her with my albino, which obviously is homozygous albino. So we got that het and that homozygous. And us knowing that phenotypically, we were able to analyze the sheds and help us kind of identify the mutations or particular patterns in albino that we can look for to tell whether any future hets are albino or not albino. Right now, she's, I bred her to, this season, I bred her to my black pastel Tofino. So she's got, I do maternal incubation, so I can't really tell how many eggs she's got, but I know when those eggs come out, some of them are going to look normal. Some of them are going to look black pastel, but I'm not going to know if they're het albino or het toffee. And so I'm planning on sending those sheds to Hannah for this semester so the students can tell me, which would be cool because
awesome.
I can kind of bring them in. They could connect with the ball pythons.
Oh, yeah.
And it's like, hey, we figured out that these ball pythons had these many hets.
Yeah, you can hold it. You can hold the snake in your hand and go, "This is the snake that this DNA sample is from."
Exactly.
That's really awesome. He said the magic phrase, maternal incubation, which is weird because like in the hobby, everybody's like they just want to shove them right into an incubator right away, and you said, "Screw it, let's see if this works."
I think that's cool.
Which is
I
awesome.
like what Sean just said.
Yeah,
This would make the world of ball pythons drop from 10,000 morphs to 100.
It would if their DNA database showed that, it definitely would shrink that morph pool.
Of course, you got to love Warren's answer, like only 22.
Yeah, Warren does not have only 22 snakes. He has more.
So Sean actually had a really great question.
Times 5.
Yeah. Are you able to tell the differences between recessive dominant, incomplete dominance, etc.?
So when we learn the genetics of a morph, it actually doesn't tell us really about whether the morph is recessive, dominant, incomplete dominant, and so on. That information comes essentially from what's already known, which is what the animals look like in different combinations. So I guess I would say it can be really interesting, though, to learn the genetics and learn that a particular mutation causes either a recessive trait or a dominant trait, and that can tell us some interesting things about how that mutation affects the cell.
Awesome. Dr. Warren Booth asked if the paper had been published. I know you said you were going to publish your findings. I didn't know if that something that's ongoing. Like there's a link that they can go check it out as you update.
So we have posted a preprint. So what that means is that we wrote our data into a paper. We posted it on a site called bioRxiv, which is where scientists post papers before they've been officially peer reviewed. We've submitted it to a journal, and right now it is being reviewed. So I can email him the link to our preprint. I'll also email him the preprint that was posted about the piebald morph, which was not done by us. It was done by another group, so he can read both of those things.
And I would gladly post any of that information you want to share with us on our Facebook page, because I know a lot of our people listening would love to go and just be able to read that themselves.
Yep, we also have a website where we describe our study, I would say in language that would be easier for a non-expert to read. So there's both of those resources available for people.
That's awesome.
So have you found with your undergrads, how many of them came into this being reptile people ahead of time? Any of them, or are they becoming reptile people, or just enjoying the process?
So none of them are reptile people. The people in my class, they sign up for the class. The title of the class does not say genetics lab in reptiles. It just says genetics lab. And
so how many people drop your class when they find out it's reptiles?
Nobody. I haven't had anybody.
That's awesome. That's awesome.
A lot of the students in my class want to go on to medical school or physician assistant school or other careers in healthcare and medicine. And so we read a lot of scientific articles related to albinism in humans or other disorders in humans. So I try to relate it to human biology, which they are really interested in. And I will say that the students who have worked in it in my lab, Chiron, you remember, so I had the student Autumn who worked on the paper, and she actually, Chiron came in to show us some of his animals. We tried to get some DNA from the mouths of the animals, and she was initially quite hesitant. I would say she would probably have described herself as someone who was afraid of snakes before then. But she did just fine, and I think that experience was a good experience for her in just becoming more comfortable with something that she has no reason to be afraid of.
That's awesome. Very cool.
I was waiting to see if anybody had any other questions.
I can hear someone driving down the street.
The bass.
Their bass through my headphones.
Yeah, I can too. I don't know if they can hear it on the
problem with
recording
living
or
in a
not.
neighborhood. Ugh, I hate it.
I'm waiting to hear the dogs downstairs.
My old neighborhood, all my neighbors would be asleep by now. There'd be no noise.
All of our neighbors were over 70 at our old house, and now we're in suburbia with families. So it's very different.
Difference, one word you can use. So my brain went blank. I had something and then it went blank. I don't know what to say now. I guess I can ask if anybody else has any other questions in our chat.
I have questions not related to the study, just general questions for them, but I'm going to wait.
No, go ahead, ask them.
Well, I feel like we're on track with this conversation, and my brain cannot go back.
Our podcast is never on track with anything. I think the one thing that's funny is, so the two of you don't understand. Most of our listeners know that I don't like ball pythons. I mean, I own some because somebody gave them to me.
Yeah, in preparation for this podcast, I actually listened to a couple of
sweet Jesus.
Well, I had no idea who you were, so I thought, what if he's very politically incorrect, and who knows? I just wanted to double check. And I happened to listen to an episode where you said you didn't like ball pythons.
I don't.
I did know that coming in.
I do. Well, I've been in the hobby long enough. I've told people before. I don't think I dislike ball pythons as much as I dislike ball python people. So you're not in the hobby. So you haven't. It's a weird group.
You did say that you appreciated that for a person who's never owned a reptile before, that it would be like a good starter animal. So that was nice that you recognize that there are people for whom it would be an excellent pet.
And what's
Hannah, when are you getting yours?
Well,
we're gonna solve
Dr.
that
Graves
problem
has this
real
plan.
quick. I'm gonna give her one of my hatchlings. She's gonna raise that up. That's gonna be her pet in her office at EMU.
I knew this was coming.
Probably not at my house. Yeah.
Love it. So what he always leaves out on our podcast is that our 10-year-old daughter, when she will walk around the shows, because all the vendors know her, so we know she's safe. I don't want people thinking we just let our kid roam.
We can always make a new one.
She always,
free range.
Pretty much. That's pretty much what vendor kids are like at the herp shows. Like everybody knows who they are and it's all good. But she'll take pictures of cool snakes that she sees at tables.
Ball pythons.
Always ball pythons.
And
she'll send them to him, like text them to him because she's allowed to have her phone if she's out and about. And he's like, why? Why is it always ball pythons?
All the snakes, it's ball pythons.
Every time. It's so great. But it's always the really expensive ball pythons. Like we have a gift. We can pick the most expensive one off the table, hands down.
My kids spent $100 on one culture of isopods this weekend.
I can't do that. I've tried a couple of times and I can't ever pull the string.
It's his money. So.
I know, but I can't do it with my money.
One of the reasons that ball pythons were a good option for us is because of their popularity. We didn't want to choose something where it would be difficult for us to collect sheds. And because ball pythons are so popular, it means that we have a lot of opportunity for people to send us samples.
Absolutely.
So the popularity for us is a real benefit.
Mm-hmm.
When Victor asked, and I know that y'all are early in this study, so I can't imagine y'all thought this far, but will you eventually do a similar study with other reptiles, or maybe look at other like boa constrictors and see if maybe their genome lines up very similar with the ball pythons? And
corn snakes is the first thing I thought of.
Yeah, so possibly. So another item that made ball pythons a feasible study for us is that even though the genome of ball python had not been sequenced at the time we started, there was a genome available for Burmese python, which we knew was closely related enough that we could use it essentially as a guide. So it would be feasible for us potentially to look at other reptiles, but the choice would probably be informed based on what the genomic resources were available for those other species.
That makes a lot of sense actually.
And I think another piece that is appealing is the fact that there's so many directions we can go with this that we can potentially keep it fresh enough that the students are seeing how the process of science actually works. Because a lot of times we have as professors, we have a challenge with kind of recreating that in our classrooms. We can do it in our labs, but recreating in our classrooms, it tends to be a little bit more difficult to kind of make things authentic. And with this project, every year we're going to have something authentic, or authentic enough that it's going to be different from semester to semester.
Oh, absolutely.
And I think it'd be really neat to get you a ball python with 4 or 5 genes in it and go, all right, guys, here's the shed. I'm not telling you what's in it.
Yes. And I will also say that when we first started this project, we went pretty slowly. So the very first semester of students that worked on this project, the only part of it they worked on, and they didn't do this for the whole semester, it was just part of the semester. The question was, can we get DNA out of snakeskin? What is the best method for doing so? And what is the quality of the DNA that comes out? And then the next semester was, is the genome of Burmese python close enough to ball python that we can use it as a resource? And we had to establish those early questions before we knew whether we could continue on as a project. So each semester, students are doing something different and building on what the previous semester of students did.
Can I ask how y'all got the DNA out of the shed? What y'all found the best method for doing that?
No, that face right there says if she tells you she has to kill you. That right
there. So the first step, and the step that is honestly the most time consuming of the whole thing, is washing the sheds. So I will tell you that people are very different in the cleanliness of the sheds they send me.
I can see that.
Some people, they'll email me and they say, I'm going to send a shed to you. I've just washed it off and it's hanging to dry in my bathroom over the towel rack or something. As soon as it's dry, I'm going to fold it up and send it to you in an envelope. And I'll get this like perfectly folded, like almost looks like an iron shed, like beautiful, clean. And then other people send me a crusty, wadded up, insect infested sock.
Something that you have to quarantine before you can bring into your collection.
I'll tell you this. So when I first started collecting sheds, I was just keeping them in my office. And I was just had these bags full of sheds, and I thought, well, they're dry, like nothing's, and it should be no problem. And then I had my student come in and I said, okay, we need to organize these. Each one needs to get an ID number. We need to enter this information into our database. And she pulled out some bags, and there were all these insects in them because the sheds had larva insects on the shed. And then they had hatched inside the bag in my office. Man. So like we couldn't even open the bag because it's full of insects. So now the procedure is, as soon as we receive a shed, we put it in the freezer so that if there are insects on it, they will die.
So how much of a shed, how much skin do you need for this to work?
Yeah, so for just a simple test, we only need a little piece of skin, maybe this big, so just a few inches square. But we do prefer to have the whole shed if possible, because sometimes students mess up, then they can go back to it. Or sometimes for unknown reasons, a procedure doesn't work. And so it is always better for us if we can have more. But if somebody was only able to collect a little piece of their shed, I would still like to have it.
Because I know Chiron's probably familiar with this. The problem with sheds is you have some snakes who use their shed as a diaper. Mm-hmm. They will shed and right away they shit and piss inside of it. And you're like, well, I assume you don't want that one. And then I'll have some who will shed and they'll dump the whole thing in the water bowl, and so you're just pulling out this soggy mess.
I like the ones that are inside out with like all this aspen when you open them. Those are fun.
They're good if you eat them early and they're still wet and you can unfold them because like an inside out sock.
Mm-hmm.
So if someone's sending it to you, what is the best way in which they can package it and get it ready and send to you?
So I want to minimize the amount of work that people do. And what I prefer is just make it dry. So even if it's really dirty, if you pulled it out of the water bowl, just let it dry before you send it to me. Because if it's wet, that can basically cause it to rot, and then the DNA can degrade. So as long as it's dry, I'm happy to wash off all the dirt, to freeze the insects so they don't pupate, all of that stuff. I just would prefer a dry shed. And if it's in pieces, I don't care either. It doesn't have to be in a whole. It doesn't have to be completely intact. If it's all shredded up into pieces, that's also fine.
Yes, and anybody listening, please do not clean it with any cleaning material, because that could also mess up the DNA as well.
Absolutely.
So
it could, although somebody told me, what did they say? Oh, they were washing their snake with like soap or something like that, and I told her I thought it would probably be still fine. So
there are different chemicals that are used if they have mites, or if there's.
Yeah, but that's not gonna mess up the DNA. That just kills them.
No, but if they have problems shedding, so we had somebody on that talked about that. It was like a soak that they would soak their snake in, and it would help with the sheds. A couple of things real quick from the chat, just because I'm trying to be the one to keep up with it. Darren Watson asked how you track the sheds that you get, and so James asked him to clarify. So basically, if Darren sends you a shed, how do you know that one belongs to him, and how do you keep the record of it?
Yeah, so we keep each shed in a Ziploc bag, and it has an ID number on it. So it gets assigned a unique ID number, and then we have a database where we know that shed 257 corresponds to such and such animal from so and so. And a lot of times the people have names for their pets. Breeders don't always name their animal, but we just have the ID number, and then we have any information they give us about the morph, the sex, when the animal shed, if they know all that information. So it's a pretty simple system, and we just keep them all. They're all in. So the small bags go in bigger bags, and so it's basically like a filing cabinet of sheds. Yeah,
that's awesome.
Yep.
So
there you go.
This is how I pack up my sheds. So you can see I got a Ziploc bag, and then I've got. Can you all see that?
Yeah, we can. Yeah, absolutely. Yeah. It's really clear too. That's awesome.
Bam, we'll even make it bigger.
Oh, I love it.
On our website, we do have a page dedicated with instructions for people for what information they should include.
Awesome. Gotcha. So Sean likes to give James a lot of grief because James is a snake guy. But there are other snakes. There are other reptiles out there.
There may be other reptiles, but the only ones they need to work with are snakes. Those are the only ones.
Well, yeah, so that's really interesting because some of the information that I've been learning in the literature about just structure of skin, it's not actually that well known for snakes. And so some of the information I'm getting is in studies in turtles and other reptiles, and that's all been really informative for snakes. So he's definitely right. I mean, I know there was a joke, but
yeah. So earlier you mentioned something about you were getting a mouth swab from a snake.
Yeah. So
can you use that?
We tried initially to get mouth swabs because we wanted to compare, is the quality of the DNA better, worse, different than a shed? And what we found is that we get much less DNA from a mouth swab. The quality is a little better, but it turns out that the quality of the DNA from a shed is basically good enough for most of what we want to do. And the hardest part about the mouth swab is persuading the animal to open its mouth. Yeah,
absolutely.
Yeah. So we kind of abandoned that as an option because it just was too difficult.
Anyone that's had to assist feed or force feed a snake knows it's not the easiest to get the mouth open.
Well, and not only that, getting a shed, that's part of life.
Well, I mean, that's invasive.
Yes, absolutely. It's a lot less stressful.
So Dr. Booth said that they use most of this Chiron kit with DTT. That's what they use for hundreds, if not thousands of samples.
Yep. Yeah, so Chiron is a company that makes a lot of molecular biology products, and they make kits that is basically like a collection of chemicals where you follow a recipe to get the DNA. And then DTT is a chemical. I'm not sure why he would use that for extracting DNA, but apparently that works for him. So we use a protocol that we basically created ourselves from some publications that is very inexpensive, and that's how we can do it in undergraduate lab course. We don't have a big budget for doing science in the classroom. So we use a procedure that uses magnetic beads where we essentially mix the stuff that comes out of the shed with magnetic beads, and the DNA sticks to the beads. And then we have a magnet where we pull the magnetic beads to one side and the DNA is attached to them. And that's how we get the DNA out.
That seems way more fun.
So cool.
Yeah.
Hey, Booth, you should do that.
Like the science experiment side of me is like, this is fun.
I like magnets.
It's like when you make your own ice cream in a bag.
Okay, maybe not that.
And actually there are, I'm sure that you can find YouTube videos of people extracting DNA in a bag too. You can do it with household chemicals as well.
What?
Yeah, I've done that before in biology lab with strawberries. You can get strawberries.
Oh, cool.
And then you precipitate it with isopropyl alcohol.
Yeah. And you can pull this big string of DNA. You can do it with your cheek cells.
So I've seen that, and I've always wondered why people use strawberries, because I feel like strawberries are so delicious, and I would have chosen like something that I would not want to waste.
I want to say I saw one that had something to do with the amount of DNA. I don't know. It was something to do why it was easier. But we also did cheek swabs and got the same result with human DNA. So I don't know. But
I think I remember you doing this. You had like a little tube and you were like, hey, look, my DNA is in this tube.
Yeah.
It's in a box somewhere.
Yeah, so if you ever do one of those tests like 23andMe or Ancestry testing, they'll send you a swab and then you rub it on your cheek and you send it back to them and they'll get your DNA out of the swab.
Yeah, shed. I assume just a shed is so much easier, and it's so much easier to collect from just the general public. Because if you're trying to do a swab, you have to send out a kit. They'd have to try and do it. And you have to hope they don't get other stuff on it.
Yeah. And another good thing about the sheds is that we can learn some information about the morph. So, for example, with the albino versus not albino, we can know whether if we get a shed and somebody claims their animal's albino, if the shed is from a normal animal or an animal that's not albino, you can tell that from the shed. So we can get a little bit of information confirming that the person had their morph mostly correct.
Dr. Warren Booth is the crusher of hopes and dreams. That's what he is tonight.
We use magnetic beads for size selection in our genomic libraries. By the way, the magnet was $1,000, so it's a fancy magnet.
Yes. So he's right that the magnets that a lot of well-funded molecular biology labs use are very
well-funded. That is Warren Booth right there.
Get it, Warren. You got money.
So we actually made our own basically apparatus. So we bought magnets online for very inexpensive, and then we hot glued them to like a plastic rack, and we did it that way because we needed.
There she is.
For every student, and so it works great. And so we also have magnets, and ours were less expensive.
That's so cool, though.
Suck it, Warren.
This is what I'm talking about, the DIY.
You got to do this shit and made a lab. Take it.
This is great.
Oh,
we haven't even had him on yet. You can't tell him to suck it yet.
I know, but it's fun.
Funny.
So
he was running earlier and he was like, I can't really type because I'm trying to run. I'm like, what is with people and exercise? It's 1,000 degrees outside.
If I'm running, something's chasing me. Y'all need to run.
I tell my students every year, if you see me running, you better follow close because there is something coming. I don't play.
Yeah.
So let me ask this. Is what you do easy enough, and I'll say cheap enough, but Warren is still running. Is it cost effective enough to be able to scale it down to, say, a high school biology lab that could afford it if they wanted to also try this?
For the tests that can be identified on by the size of a band, as we talked about earlier, the answer is yes. There are so the type of mutation that an animal has in some ways dictates the type of test you need to use. And for many types of mutations, yes, it could be used in a high school biology class. There are some that are going to be more complicated.
So in the future, I see high school teachers being able to purchase a kit
That comes with the shed or with
that you guys have put together for them that says
you're giving them more work now. They don't want more work.
This could be 5 years down the road. It doesn't have to be now. This is a good undergraduate project for someone to put together. Put together kits that could be used in local high schools.
Yeah. So I described this project to a friend of mine who is a vet, and he said, "Oh, you could sell that as a kit." And so
yes,
too. But I don't have plans to start a company of ball python high school kits. So
Girl Scout,
I'd be happy to provide someone else with all the information they would need too.
The Girl Scout troop leader in me is like, how can we fundraise? And this is how you fundraise.
The business part of me is like, I know a lot of people with ball pythons, and I can just get the information, and I can start a whole business where I just sell kits to other science teachers.
I mean, there you go, James.
And we never have to use another strawberry ever.
And
again.
you can donate a percentage to their research. A percentage of every kit you sell gets donated to their research.
Yeah.
Totally worth it. Then I get people to just map the genome of my snakes. I would love to see a poster of like the morph and then the bands, like the little picture of the bands that go with each one.
I did. I posted. So there was somebody. I don't know his last name. He's known to me as Steve P. from someplace in Idaho, who wanted me to test his animal for lavender albino. And I posted his test on Facebook, and I put the picture of the gel and said, here's your test. Your animal is not heterozygous for lavender albino.
Crushed his hopes and dreams.
Day.
Here's a picture to make you cry. Now have a good day.
Oh, that's so funny.
Oh man. There's so many. You could just imagine like this test going in such wrong ways. It's like those ones where someone doesn't realize they're adopted, and then they do the test and they realize their DNA doesn't match their parents. So this person spends $10,000 on this het whatever.
You just got really dark there,
then
man.
They spend $10,000 on this het whatever, and then they send in the test, and it goes, "Oh yeah, it doesn't actually have any of that in there."
Yeah, so actually that sort of happened a little bit with Chiron. So in Chiron's case, and he and I maybe disagree about this a little bit, but he has a snake that was sold to him as het albino, and he has another snake that was sold to him just as normal. And the one that's described as het albino in my test does not look like it's het for albino, but it just so happens that one of his other animals, this normal animal that was not sold to him as het anything, looks like it's probably het for albino. So he kind of lost one, gained one.
It's kind of like when you got a snake in the mail.
Normal was a pet store special, not Petco. We have a local pet store here in, actually it's in Saline where I got him. And so it just looks like a regular normal. But apparently he's het for albino. But the clown that I got that is supposedly, and I just wanted a clown. I didn't care whether it was het albino or whatever, was supposedly het for albinism. But apparently he doesn't have this allele. So
have y'all figured out any percentage of error in these tests, or is it pretty accurate every time, or is there a?
So that's a good question. So I guess my feeling is when I do the test, it's essentially always accurate. I do sometimes have a concern about my students doing it in the classroom because they don't always follow the instructions correctly.
No, that can't be true. Students are amazing.
What?
And I will say that I have given the same students, different students the same animal, and they've always come back with the same answer. So I think that it is mostly accurate.
Now that's a good idea. Having them all work on the same one to make sure they're checking each other's work faithfully.
Make that their final. Give them all the exact sample of DNA, and then whoever gets it wrong fails the next year.
Gosh, James.
Doing something that I don't know if this is really like that I should say this as an instructor. So I can see the students' grades in their previous classes before I start the semester. And I noticed that students who have higher grades in their previous classes are ones that tend to, pay attention better and follow instructions better. So I've considered, for example, giving one shed to 2 different people and specifically always choosing a student who had higher grades and a student had lower grades so that I, make sure everybody's getting the experience, but at least getting a stronger student on that.
So in elementary school, we call that differentiation, and it's totally fine. We do it all the time. That's all you're doing. You are ability grouping and differentiating your projects.
You just give a fun words that make it sound like you're doing something fancy when in reality you're just.
I do that all the time in 5th grade.
Yeah, we hammer that home in education programs too. How are you differentiating?
Yep.
Let's see. One of our they asked. I'm gonna read this out loud. I'm see if I make sense of it as I read it. So in blood pythons, I hear matrix, which is heterozygous, and matrix, which is heterozygous. The homozygous form is ivory. Now I hear hitting the homozygous form is exceedingly hard to get when it should be a 1 in 4 chance of hitting it. Now, is there something else going on in the allele that makes that harder to make them, or is it just that bad of luck?
So I'm not familiar with this exact situation, but it could be that the homozygous animals have a higher rate of lethality during embryo development. And so they just don't always hatch. That's possible. So if he's basically getting a lot of dead or undeveloped eggs, that would, once you add those together, would make it 1 in 4. That's probably what's happening. I think without more information, it'd be hard to say for sure what's going on there.
Yeah, I'm not sure. I know our buddy April will probably be able to answer that much better than I could. She breeds bloods. I know there are some things in certain snakes that the motley gene and boas, if you get a super motley or a homozygous motley, they tend to die fairly early, somewhere around 2 or 3 years old. I know that the homozygous version of jaguar carpet pythons are solid white, but they are dead. They don't actually survive being born. I know there's several genes where that happens. And if it happens early enough, obviously you would never really catch it because you would never see the dead animal to prove it.
Yes, and typically morphs that are completely white like that usually have, at the molecular level, a pretty severe defect in terms of there's a lot of cells that are making the wrong decisions about either where they need to be or what type of cell they need to be. And so the fact that it's called ivory, so I'm presuming it's an all-white snake.
I think so.
That also makes me think that this scenario is more likely.
Awesome. All right, so James, when you started this podcast, you agreed that it would not just be an interview.
Yeah, but
podcast.
this is different.
This is different.
I wanted to know this is an interview. Like I didn't plan the other stuff we normally do because there's a lot of interesting stuff here that I know people wanted to hear about, and that this whole thing, if done and people really buy into it, could definitely change the hobby. I think for better, because there are a lot of people out there that sell there's a lot of people out there that sell hets that are not hets. And if they can send them some sort of certificate back going, look, this is what it is, and they can sell that with a snake, that makes your $200 snake now maybe a $2,000 snake, because I can prove, look, this is it. You are getting this.
But you also have it on the other side of the spectrum where your $2,000 snake may only be a $200 snake, which is why Hannah's having issues getting some sheds from some of the breeders.
Yeah.
And it is what it is. I mean, I think the reputable people will want to know what they have.
Oh yeah.
I mean, there's a lot of stuff going on with genetics lately. The ability to test your animals for what is the virus I can't think of that's killing snakes?
Nido.
Yeah, the way they have those tests for Nido, I think those are amazing. You can test an entire collection for Nido. I think and especially like what they're doing, this test is fairly inexpensive, which is for a genetics test is amazing, because I think 15, 20 years ago, anything genetics would have been $20,000 to try and do, and y'all can do it for fairly cheap.
So I will say, so the reason that genetics tests, like let's say you were planning to have a baby and you wanted to be tested to know whether you're a carrier of certain genetic diseases, and that would probably cost you several $100s depending on exactly what you wanted. Most of that money actually goes to pay the humans who are doing something with your sample in the lab, manipulating it. In our case, the reason it's less expensive is because we have students doing it who are learning the genetic techniques, and so they're not being paid. It's part of their education. So that's
they're being paid in experience.
Yeah, yes. I mean, technically, they are paying to have the experience.
That's right.
but it's not like you understand.
Okay, so my question is, and this has nothing to do with your research at all, and all to do with you. What podcasts, if any, do you listen to for fun or in your free time? Are they science related? Are they reptile related? Are they?
Well, I'm gonna go with Hannah. They're probably not reptile related.
Are they education related? What podcasts do you listen to?
So I can start. So some of my favorite podcasts, they're not really education or certainly not reptile related. One of my favorites is 99% Invisible, which is a design podcast, and design is broadly interpreted. So it's about cities and to call it about cities. It's just about like human made design things and how they can be better. That's one of my favorites. I also like the podcast Criminal, which is in theory about crimes, but it's not really a true crime podcast. It's about all sorts of interesting things involving crimes.
That's cool.
Robert
Gray.
loves true crime podcast.
I'll listen to a few.
Yeah, I know. Because you always talk about you listen to true crime. I can't get into true crime podcast.
Yeah, I mean,
I listen to what James considers really boring podcasts.
Yeah, you listen to podcasts about quilting.
I think it's cool.
I listen. I feel like if it was a quilting podcast, you would want to see what they were talking about at the same time.
So a lot of the quilting podcasts that I listen to actually have to do with the history of quilting.
Oh.
So like there's a couple that have dealt with like quilts from the Underground Railroad and all the research done with those.
I remember when those were a really big deal.
Yeah. So it's stuff like that on those podcasts, or like there's different organizations that make quilts, like the Quilts of Valor make quilts for veterans. So it'll be like, this is what's going on in the world today type thing. And this is what we've done. And he makes fun of me for it, but it's okay.
Sean.
So Chiron, what kind of podcast do you listen to?
So my favorite is this podcast called Throughline, and it's just because I'm a history nerd as well, and so podcast does a really good job of kind of delving into the history behind like a ton of things. Right now, they're looking at they're doing this series looking at capitalism, but the other one is Hidden Brain. I like that one. So I mean, I guess you could say they're educational, but they're kind of fun. They're not related to what I do specifically in my work. So
that's cool. My daughter and I.
That one Throughline.
Oh, look. Robert's adding them to his list as we're talking. So my daughter and I actually recently listened to a podcast called Frog of the Week. Oh. And it's like 2 to 3 minute little clips. They come out once a week. We actually listen to like everything that's been out so far in the span of like 45 minutes.
We
binge listened Frog of the Week in about 45 minutes in the car. But what was cool about it is, okay, let me rephrase that. What I thought was interesting is like episode 1 or 2, my daughter was like, I just don't know. There's something about her voice, like, because she's very relaxed and calm, and she doesn't really get super excited. It's very informative. It's almost like when this is going to show my age. When you go to a museum and you get headphones and a little cassette deck and you walk around and you do your own personal tour. Do they still do this? I don't.
Yeah, but now it's probably on your phone. It's
probably a podcast on your phone now.
Yes.
But anyways, but that's what her voice sounded like to me. But it was such a neat. I like frogs, so it was a really neat podcast for me because it's Frogs of the Week. And every week there were frogs that they were talking about that I had no clue even existed.
Which would be most frogs.
But it was really cool. It includes sounds of what those frogs sound like in the wild and little facts about them. And that one was fun.
So there's another faculty member in our department who teaches the herpetology class, and I'm going to tell her about this podcast. I bet she might be interested in doing some type of assignment, where students maybe have to listen to some episode and answer some questions or like investigate one of the frogs we talked about.
So you have another professor that teaches a herpetology class. Have they tried to reach out and tie into anything you're doing or connect? I mean, I imagine what you're doing has to really interest them. I mean, I guess if they're field herping, it probably not as much.
They have answered my really naive questions about reptiles.
I feel your pain.
But and they did put me in touch with someone at a local zoo who I asked them questions about DNA extraction too. But I would say the person who teaches that class is, I would describe her as really interested in reptiles in the wild and less interested in captive reptiles.
And I think that's always been one issue with the hobby. The college side of it, the intellectual side of it, and the hobby side have not always seen eye to eye. So it's very interesting seeing you doing a study at a college level, which is very much based in the hobby. You know, you're looking at these morphs.
That shows my naivete when it comes to reptiles in general.
But you know that.
But see, I disagree with that.
Oh, what's that?
I disagree. I mean, it may have started out as you being naive and not knowing about the two crossing, but in reality, it's almost like you're bringing the two together.
Yeah, I think what I bring is the eye of the geneticist to make hypotheses about what genes might be causing some of these morphs. And right now, I'm planning to write a small paper where the sort of take-home message of the paper would be, hey, geneticists, look at all these ball pythons out there. They're this really interesting resource for understanding the genetics of color in reptiles. Here's what they look like. Here's some ideas about what the genes are. No one is studying them. They're this resource that you didn't know about.
Pay no attention to the silly names that they gave them.
Yeah. Yes.
So Sean says, so hobbyists can't be intellectual now, James. Sean, you run a reptile expo. You know that not all hobbyists are intellectuals. Okay, I'm just saying. We're going to tell the truth.
I went to a reptile show here in Michigan for the first time, and I was surprised.
Oh, goodness.
Well, I was surprised you could also get like your body jewelry and your like piercing supplies also at the reptile show.
We're a weird group of people.
Yeah, we are. That's putting it lightly.
It's loosely defined as Reptile Expo.
Yeah.
It can be very interesting being at a reptile expo. Warren says he integrates undergrads through postdocs into all my reptile work. Our new Python phylogenetics work was undergrad driven.
Come on, James.
I know it.
Come on.
I should look into that phylogenetics. So basically, he's saying we're trying to figure out which pythons are related to which other pythons.
Yeah, he said also the parthenogenesis work often includes undergrads. For example, the balls and retic pythons, parthenogenesis paper. Yeah, he's done a lot with the partho stuff. I find very interesting. And the most amazing part that I found, and I thought it was funny because Warren, I don't know how much time y'all spend on Facebook in any of the reptile groups, but it gets very interesting on Facebook when you talk to Facebook experts about reptiles. So Warren was talking about parthenogenesis, and someone told him he didn't know what he was talking about, which I thought was hilarious because he wrote the paper. But that parthenogenesis actually happens way more often in ball pythons than anyone actually thought of. So it's very interesting for something like what you're doing. Someone could have a baby in a clutch, and one of them could be a partho baby and not actually be het for what it should be het for.
Yeah, that's interesting. Yeah. Actually, there was recently someone emailed me and said, I'm sending you the shed, and by the way, I think that this animal might have come from parthenogenesis. Can you test? And I said, I might be able to, sort of depends on the genetics, but I can keep that on my list of student projects. And she said, okay, great. So I should probably, I should look into how common it is. I hadn't realized how.
Yeah, Dr. Booth, he had a lot of them. He would get, and that's when he found out that it was very often that you would find ball pythons that were through partho. So that's one area where someone could be selling a pos het albino. Or het albino. Should be het. One of the parents were albino, but it comes out it actually was a normal. It never got an albino gene.
So Dr. Graves was saying that he's got his male, he's got his female, he's got some eggs, and then he's going to have some of Hannah's students go through the sheds. I mean, there's a possibility that he could have something like that pop out of one of his clutches.
And actually, and one of the things that I forgot to mention this, but last year and the year before, one of my females, she had both years she had a surviving clutch of 5. So one year was 5, the other year was 6. And in the first clutch, 2 of the babies that came out were spider. Neither of the parents have spider.
Huh.
The next year, a third one came out spider. And so one of the things that I'm planning on doing is having Hannah look at those sheds and determine if the father, who I think is the father, is actually the father, because just on the surface, it's like, this shouldn't have happened.
Yeah.
And so the genetics test our testing works out in that way as well to kind of confirm not only partho, but, paternity.
I think that's really, that to me, I think is really cool.
I just want to say Warren also said that all of his papers on what he wrote was on his website, booth-lab.org.
Thank you. I will check that out. I do. The paper of his that I'm most familiar with is the one about the sex chromosomes in boas and pythons.
And boas, yeah.
And there's a lot of work that goes into earning that doctor title. So I don't ever know when I should use it and when I shouldn't use it. So I will just call you Warren.
Yeah, Dr. Booth.
Yeah, I think that snakes can store sperm for a long time. I think that's true. And same with turtles. They can store sperm.
He said a recent paper showed storage for 6 years. That would make breeding miserable.
But here's the thing, though.
I saw someone who had a clutch. They just had a female lay a clutch that they haven't paired in 3 years. And when they had the babies, they were definitely from that male from 3 years before.
But has your female that threw the spiders ever been with another male?
Or with the spider?
The first year, she had been with 2 other males, but neither one of those males have a spider either. So I'm in the second year, she was just with that one male.
And if I remember, spider's a incomplete dominant trait, right?
Yeah.
Yeah. So I mean, they would have to either be or not be. So that's the weird thing there. Although I mean, well, it can't be. I would say you could guess for a random mutation, but a random mutation doesn't happen 3 times in one snake's babies. So that just would be weird.
That's cool, though.
Yeah, that's
interesting.
That's one of those situations where an answer for something may raise more questions.
Yeah.
All right, so we talked about podcast stuff. Do either of you, and you don't have to tell us when you watch YouTube, but do either of you...
We all know when people watch YouTube. It's when they poop.
No, I watch YouTube just to watch YouTube.
Pooping every is the time to watch YouTube.
Every week. Oh my gosh. Just because you want to watch it. Do you watch anything on YouTube? Like, I don't watch a whole lot of YouTube. I have more time to listen to things like podcasts than I do on YouTube to watch things.
They're busy working. They're actually
I know. Do y'all watch anybody particular on YouTube?
I'm like you. I mostly just listen to podcasts. Occasionally, I'll look for something on YouTube if I'm looking for instructions for how to do something. Like, I need to disassemble my vacuum cleaner, and now I need to find a video of somebody showing me how to do it. So it's things like that.
I've got to do that.
I'm changing the shocks on our washing machine tomorrow.
Oh, I actually just watched some videos about washing machine repair, doing a similar repair myself.
Yep. The parts are cheaper on Amazon is what I have found. Just a heads up.
YouTube has been really good with that DIY. Like we just recently had we had our wax ring go out in one of our toilets. And so it was like, well, I guess I'm pulling up YouTube to check out a video on how to replace this thing and what happened. Because at first I didn't know what happened. We just had this water on our floor, right?
I started an entire construction company years ago, learning how to lay tile on YouTube, and it just grew and grew from there.
It's awesome.
Did you learn how to lay tile while pooping on the toilet?
No, James. No. Not everyone watches YouTube on the toilet. Nope.
When else are you gonna watch it?
When you need to learn how to do things like take apart your washing machine or flip the door on your dryer so it opens from the left instead of the right, which I also did and felt super proud of myself for doing because you weren't home.
So I think Sean. So Sean asked, "Are there anything published yet on the lemon frost results?" I don't think that was y'all's, right? That was somebody else.
Right. That was somebody else, and that actually was published just a couple of days ago. There's actually some. I think if you Google lemon frost leopard gecko, it'll probably come up. It's in the journal. Is called PLOS Biology, PLOS Biology, and it's received some news coverage, I think, in Scientific American and maybe a few other places.
Cool. So Warren said, spider blackheads look normal, so a male could look normal but make spiders.
Yeah, but the 3 males in question, you would definitively know. One of them maybe. So one is the clown. One was a pastel inchy het clown, and then one was a banana lemon blast. So it's possible the lemon blast could have had spider and you couldn't really see it, but
yeah. So Sean said the lemon frost gene causes fatal tumors.
Okay, so I actually Googled it when she said that, and I have, because I get the Reptile magazine. Well, you get the subscription, which I need to change the address for, by the way. Don't let me forget. But I get the email updates, and that was one of the things that was posted in the email update. It was talking about the tumors and the cancers and things like that.
Yeah, so it's really interesting. So you guys probably know that in reptiles, there's 3 different types of color cells, whereas mammals, we just have one type of color cell. And in mammals, our color cells can cause tumors, which are known as melanomas. And in these leopards, they have a tumor that's formed by a different type of the color cells. So it's kind of like a melanoma, but it's formed not by the cell that's making melanin, but by the cell that's making a different color structure. So it's actually really interesting.
So I think I actually just found the paper you were talking about on bioRxiv.
Yeah, it's pronounced bioRxiv. So that's the
okay.
And it's since been published in an actual journal, and there might be a link from bioRxiv.
We can share that over there on the
that's
group.
I was going to share it on the group.
Like enter stuff into the chat. I'm not totally sure if I.
Yeah, you should be. I can't because
okay.
My computer's not actually.
So Sean was asking because we talked earlier about some of these fatal genes. He was wondering if tests like this could be used to help identify fatal genes. I guess you would need to know ahead of time and know what marker to look for.
Yeah. So in a sense, yes, if they were always, well, okay, so if something always caused the animal to be fatal and it was dominant fatal, of course you couldn't get animals to look at, but if it was something that was homozygous fatal, yes, you could be identifying that.
Gotcha.
I didn't understand that.
Okay,
so
sorry.
There's
no. But what I'm thinking of is I've talked to someone about fat tail geckos, I think it was, and you can't breed this type of fat tail gecko to this type of fat tail gecko because the babies will never make it. So I would assume maybe with a snake, it's similar. Like you can't breed this species, this morph to this morph, or none of your babies make it.
There's some where the fatal is it's in formation. So yeah, so you would never form. But like we're talking about something like the super motley boas or the codominant or homozygous motley boas.
Mm-hmm.
They can reach breeding age technically, so you could still get one later in life that still carries a fatal gene that's going to cause them to die early.
But you would have to know which genes were the fatal ones in order to say, yes, your snake has this, or no, your snake doesn't, right?
Yes. In order to like recognize that these fatal genes even existed, you would need to have some indication that animals were dying.
Right. Like you would have to have enough people say this.
Yes.
Pink gene. I'm trying to think of something that's not a ball python morph.
Yep.
Good luck. Every word is used as ball python morph.
Like you would say like this pink morph.
Yeah.
Dies whatever.
In the case of these lemon frost animals, they don't die during the embryo. They grow up and then they get a tumor as they're older, and so that's how you can get information from them, because they can die early, but they're not dead from the get-go. I put the link in the private chat. I don't see a way for me to put it in the.
I'll post it.
Okay.
I'll post it on our Facebook page. But Sean said the same with the lemon frost gene, that that's why he doesn't allow people to sell them at the shows, because you can sell one and then it will die later.
Right.
Which unfortunately there are some people in this hobby. I mean, with any hobby, there are shady people knowing that this animal will die early. Let me go ahead and get my money now.
Okay. So that was my next question so it basically, like, there are some genes out there that will significantly shorten the lifespan.
Yes.
Yeah.
Okay. So like chameleons that only live for 5 years, you're buying an animal knowing it's only going to live for 5 years. Whereas leopard geckos may live for 20 years, but you could be buying a genetic leopard gecko that's only going to live for 5.
Yeah. So like Robert has a motley boa. That motley boa is healthy. But if I were to breed that motley boa to another motley and get a solid black boa, it'll probably die around age 2 or 3.
Oh.
So yes, I could sell it off.
So now I understand the question and the not, because that's why I'm like, if it's fatal, it's not going to live. But I didn't understand that they would just live for a shorter lifespan. So I get that now.
Yeah, there are some genetic traits that don't become fatal until later in life. So be fatal prior to breeding age or after breeding age. But yeah.
Okay, that now his question makes a lot more sense.
Ta-da.
We got there.
That's actually a big controversy in human genetic testing. There are certain genes you can learn that you have that might indicate you're going to have a shorter life or have certain diseases when you're older. And so there's big ethical discussions about should we inform them that they have that if they didn't specifically ask about it, because they may want to know that information or they may not. You could imagine that some people would and some people wouldn't.
Yeah, some people will take it as, all right, now I'm going to go do everything I couldn't do if I don't live this long. And some people take it as being depressed because now they know they're going to die early. And
yeah, or some people might just use it to, plan to make sure all their finances and everything are in order so that they...
those people are responsible. Forget those people. If you're dying early, go jump out of a plane and go do whatever.
Right.
I'm not allowed to jump out of planes.
Well, if you're going to die, you could jump out of a plane.
That's not cool.
You can't do it beforehand, because if you're not supposed to die early and then you die because the parachute doesn't open, then I'm left alone. And I'm
not allowed to skydive
until I die.
Until James dies.
Yeah.
Yeah.
Because a parachute may not open.
Okay. Moving on.
So like Robert said, we have reached 2 hours.
We have.
We've covered a lot of stuff.
This has been awesome. Yeah.
I'm extremely interested to see where this moves on from this point on, because like I said, as a hobby side, and I know Hannah definitely does not come at it from a hobby side, but from the hobby side, I see where this could be a huge thing just to clear up a lot of confusion and purposeful confusion that has happened in the past in this hobby. And I love the idea that we could go, all right, this is definitely what it is. I don't care what you say. Genetics says this is what this thing is. You can call it whatever imaginary name you want to, but it's the same thing as what that guy's selling on his table. They're the exact same snake. And you don't have because there was somebody, we saw somebody in a group the other day post something that said they had a brand new gene, that they were the only one that had it, and they were going to make tons of money. And it was the same thing, something else.
That guy has done that about 5 times.
Yeah.
And he bought some normals and pastels, and he's got a couple of dinkers that are imports, and he thinks everything's a new gene, and then when people tell him it's not, he starts cursing them and then starts calling them thousands of times. And that was
well, I cannot thank either one of you enough for coming on. This has been super awesome. I might actually retain some of this information, which would be great. Travis Wyman will be so proud.
When we were having dinner before this, Katie was like, I don't really know what I'm going to contribute tonight.
Yeah, no.
And you can tell
my head hurt thinking about the conversations that were going to be happening.
That's
tonight.
why it's useful to have you, because I could talk on a more genetic level, but I would talk over stuff that you wouldn't understand, and I would assume that everybody understands it because I took genetics classes in college. So I would assume
not everybody had to.
No. And I still hate fruit flies.
Well, thank you so much for having us. I really enjoyed it. And
this has been a blast, guys.
I'm so glad.
Really fun. I like getting all your questions and answering all these questions. And I need to, I think, learn to give answers better that are more appropriate for someone who doesn't have as much knowledge about genetics. So that's been really good practice.
Well, let's be real though. Your students probably have way more knowledge about this than I do. So you're probably used to speaking on their level.
I've met undergrad
not so much mine.
I have a variety of students, and some of them do and some of them don't. So
I would second that.
Look, I've met undergrad biology majors who were just there for nursing school. And so when you get into something like this, they're like, I don't know what we're doing.
Yeah, I have taught 5th grade for several years, and this upcoming year I will be dropping down to 3rd grade. So
that's too young.
It's going to be fun.
You can't yell at 3rd graders. I can yell at high school kids.
Excited. Victor, I think that this being able to genetically talk about the markers and the morphs, I think it will bring up some controversy.
But the problem is it'll bring up controversy, but it really won't. Like the great thing about science, and it's always an argument when people want to say something doesn't exist. In science, it either is or it isn't. Those are the options. Like, you can disagree with it all you want and you'll be wrong. But
like our favorite Texas Black Panther.
Oh yeah, the Black Panthers don't exist. The group where everybody kind of said Black Panthers are real, and it's not even a thing. But yes, so like they have the picture of these markers. They can go, look, it is this or it's not this. Now you can get upset and argue it, which will just be wrong.
Right.
So yes, there will be controversy, but the controversy will be between people who literally barely passed high school biology, and now they're trying to tell doctors why they're wrong, which I always find hilarious on Facebook.
And I agree with Sean Gray. Controversy
100%.
is needed, and it makes for great conversations like this was. So Hannah, if people want to get a hold of you, what is the best way to do that?
They can email me. Can I put my email in the chat, or should I?
Yeah.
We'll take it and we'll put it in
Yep.
when I post this. So we'll get it from you and we'll do that. And
because I've already posted the leopard gecko and tagged Sean Gray so he can see the article.
An address for where they can send sheds to, or wherever a website you want to send them to read all that.
Okay. I will send you an email immediately after this with a link to our website and a link to the publications. And I'm probably also going to CC Warren Booth on that because he said he wanted that information as well.
Absolutely. That'd be great. Sounds great. And then, so we'll get that out to our listeners in case they want to reach out to you. And then Chiron, if people want to get a hold of you.
Yeah, so I've got a couple of Facebook pages that I should probably check more frequently. We were talking about YouTube. Started them, I thought it was a good idea, and then realized that wow, a lot of things are happening at work right now. Kind of hard to keep up with Facebook, but I can put that in the same with Hannah. I could put that in some of that stuff in the private chat.
Perfect.
As far as like Morph Market, I'm on Morph Market as Wesley Ball Pythons. Wesley's my middle name because people always mess up Chiron, which is why, like I said,
is it Chiron?
that you all got it right the
and
first
we're in the South, so yeah, Chiron would have definitely been how it went.
Yeah, my cousin used to tease me about that.
Oh my goodness!
But
like, I'm a high school biology teacher. I've seen way worse names that I had to try to get on the first day. There's only 2 vowels in that name. We're doing pretty good.
Yeah, there's a lot of times where I will call students by I'll say Mr. So-and-so, and I'll call them by their last name because I know I can pronounce their last name. And then I'm like, so how do you say your first name? Teach me. So, but yeah. All right. So Robert, if people want to get a hold of you.
www.lsreptilerex.com.
Why? Every time, James. Every time.
I don't want my brand to be tainted with that dumb shit.
No, James.
Yeah, that's horrible.
We don't want your dumb shit.
It comes along with the podcast.
We have actually, our listeners are maybe like messaging us to make sure that we are like well and healthy because we were very professional tonight, guys.
Yeah.
You done messed up, AA Ron.
Oh my goodness.
Anyone that's a teacher definitely has to go watch that from Key and Peele, the substitute teacher clip from Key and Peele.
Mm-hmm.
That's one of the best ever.
Oh my goodness.
If you want to get a hold of us, or
Yeah.
for me, it's simply _serpents on Instagram or simply serpents at
Talking to the mic, James.
I am simply serpents at simply serpents on Facebook. God, it's been forever. Simply serpents on Facebook. I will have rainbow boas. Hopefully for sale in
They are bitey.
2 months. They are bitey. They bite like hell. If you want to get a hold of us, it is the Reptile Gumbo podcast on Instagram, Facebook, and at gmail.com. That's where I was getting confused.
So you two stick around. We're going to make this not a live broadcast anymore, but don't go anywhere yet.
But thank y'all. I want to thank all of our listeners for the questions. I want to thank the several PhDs that we had tonight all over the place. I feel smarter by being
acquaintances.
In the same.
They might not
same
call
chasm.
you a friend.
One of them's a friend. He sent me a snake. He's my friend.
I thanked him for pretzels that he made. You did not.
Travis Wyman sent me a snake.
He sent me a snake for you.
That's true, but it's
you didn't have to drive to the hub and pick it up.
I've met the guy, so there you go.
Anyways, thank y'all for coming on. And that is it. Good night. Bye-bye.